లైవ్ నేను చెప్తాను రండి.
Welcome to Zoology webinars of Model School for Junior Inter. Good morning, children. In the previous sessions of Zoology, Miss Jhansi ma'am explained us about various types of tissues in animals. While explaining, she was referring to germinal epithelial tissues, isn't it? So what is this germinal epithelium? Where does it come from? And what is there in connection with the organ and organ system evolved? Isn't it? All these questions will arise when you are learning about all these things in detail. Today, Madam will enlighten us about the fluid connective tissue, lymph, and also about how in an animal kingdom, levels of organization evolve. Please make a note of all these terms and the points in a running notes maintained by you all, especially for the webinars. Okay, why? Because unless you know these meanings and understand what exactly the term defines, it's highly impossible you all to know the characters, especially when we are dealing with the classification of invertebrate and vertebrate. Each and every teacher will be explaining you about the characters. They just say diploplastic, coelomate, and all those stuff. So unless you know the terms, how they are evolved, you cannot learn the characters and remember those things. Okay, so please make a point that you start writing the notes while ma'am is explaining. Today we have Jhansi ma'am with us, and she is PGT from Munipalli from Sangareddy districts. Please take over the session, madam. Good morning, madam.
Jayalakshmi ma'am, good morning, and thank you for the introduction and summary both, ma'am. Thanks a lot. Let's not get waste with our time. Let's not waste our time. Let's get started. Yesterday I stopped with RBC. Today, let's continue with fluid connective tissue. Slideshow. From beginning. Ma'am, can you see? Hello?
Yes, ma. Yes.
Ma'am, can you see my slides?
Yes.
Thank you, ma'am.
Proceed. Yeah.
Yes, students. Yesterday we finished with fluid connective tissue introduction. We finished with RBC and the plasma, that is a matrix of blood. Today let's get started with white blood cells. That is, these are nothing but the leukocytes of our blood. WBCs are also called as white blood cells, or they are also called as leukocytes. So these white blood cells are called white because they don't have hemoglobin. We have studied yesterday in RBC that RBCs are red in color because of the presence of pigment called hemoglobin, but these don't have that hemoglobin. Hence, they are white in color. If you talk about their shape, they are spherical in shape or irregular in shape. So this is the difference, one of the differences that you find between RBCs and WBCs, right? One more point regarding RBCs and WBCs is that, we find very big nucleus over here in WBCs, whereas we don't find that RBC having nucleus. This is another striking difference between WBCs and RBCs. Now, as we go through WBCs, we try to learn about it, we'll come to know other differences too. Now, what is diapedesis? WBCs have the quality or the nature to come out of the capillary walls. You know, WBC is a part of blood. Blood flows through capillaries, blood vessels, and then blood vessels ending with capillaries. So these capillaries have minute spaces between them, and these WBCs, they exhibit amoeboid movement. So slowly they come out of these capillary walls and enter the tissues. So this movement of WBCs through the capillary walls and entering into the tissues is called as diapedesis. Now, number of WBCs. Yesterday we have learned about RBC. Formation of the RBC was erythropoiesis. So here formation of WBC is called as leukopoiesis. Then for leukocytosis, see, WBCs help especially in protection of our body, which we have been learning since our 7th class onwards. So whenever there is a slight infection in your body, that time the number of WBC actually increases, and that condition is called as leukocytosis. On an average, the number of WBCs per cubic millimeter is only 6,000 to 10,000. Whereas RBCs, their number was around 5 million in case of males and 4.5 in case of females. See, where is that million and where we are talking about here? Only 6,000 to 10,000. The number is very, very small. So whenever there is an infection, there is a slight increase in the number of WBCs. That means your body is fighting that time with the germs. So that condition is called as like leukocytosis. Abnormal increase in WBCs, that condition is called as leukemia. As we have seen yesterday in RBCs, polycythemia, which is nothing but a type of cancer. In the same way, leukemia is also a type of cancer. Less number of WBCs than the average level that is needed, than the range level that is needed, that condition is called as leukocytopenia. So here is a slide which shows actually the number. See here, in normal blood, you find that there are only 4 WBCs that you can see, but here the number is more. This is abnormal condition, which is leukemia, that nothing but cancer. Types of RBCs. We have learned about WBCs. RBCs are only of one type which we have studied, but we can divide WBCs into 2 types based on the presence or absence of certain substances called as granules. Granules are present in cytoplasm. So what are these granules? Granules are nothing but they are enzymes that are helpful, that do the work that particular WBC is supposed to do. So those enzymes are present. When enzymes are not present, you can't see those granules, those dots. So those WBCs are called as agranulocytes. Granulocytes, again, we can divide them into 3 types. Granulocytes are once again divided into 3 types. Now, what type of division or category that we find is, it is based upon the type of stain that these cells can take in, the type of dye, dye that these cells can take. Depending on that, we categorize them into 3 types. So basophils, acidophils, otherwise eosinophils, and then neutrophils. So depending upon the kind of material that they take, I'm sure you would have seen cells under the microscope, right? So you find different colors of the cells appearing sometimes in pink and blue color like that. So those are all because of the dye that we use, the stain that we use to stain these particular cells. So similarly, these granules have the capacity to absorb some type of color. Depending on that, we divide them into these 3 types. So first one over here in front of us, we have basophils. That means these cells can take up basic dyes, alkaline dyes, basic dyes. So here you can see nucleus is either 2-lobed or 3-lobed. Here in WBCs, nucleus is of various shapes. So we call it as actually polymorphic nuclear condition. Many forms. Polymorphic nuclear conditions. In RBCs, nucleus is not present only. So that is enucleate condition. So the percentage that these basophils make up actually is only 0.3. It's very, very less, 0.4%. Very less amount of WBC of basophils are found in blood. Anyways, let's talk about its function, basophils function. Basophil functions, they especially help in the production of heparin. That means these do the job of, or they supplement these mast cells. You remember this particular slide which I showed you in areolar connective tissue? One of the cells that we have studied over here is mast cells. And mast cells secrete heparin, anticoagulant, that keeps our blood in liquid condition without forming clots. So this helps in production of that. That means basophils, they take up the job of substituting these mast cells whenever it is needed. So apart from heparin, these also secrete vasodilators and vasoconstrictors. So this is a function of it. Moving on to the next, acidophils or eosinophils. So these, as I said, that they take acid dyes. So their percentage is around 2.4. Their percentage in total WBC, if you look at, right? Again, here also these are 2. You can see it is bilobed conditions, right? In the earlier case here, the granules are very large. They are very large, very few, and irregular in shape. So here also you find granules are very large. This is again a bilobed nucleus that is found over here. What about the function of acidophils? Acidophils especially take part in removing antigen-antibody complexes, and they play a major role in allergic reactions. So what is this allergic reactions? Let's try to first understand this. You can find sometimes, even you would have personally experienced this, or would have noticed, among people, this condition. Suddenly, if they try to take some kind of medicines, or due to any reason, you can find rashes on their body, right? So that is, you might have heard about people saying it is because of allergy. Allergy is a condition where actually our immunity, our immune system tries to doesn't recognize our own body cells and start attacking them. So that condition is called as allergic condition. So in that allergy process, these acidophils play a major role, right? Next one, they help in removing this antigen antibody complexes. I think I already shared with you what are antibodies. Antibodies are proteins which are produced by B plasma cells, right? Plasma cells derived from B lymphocytes. So these antibodies, proteins, they take up the antigen and they form antigen of what? This is again protein that is found on pathogens, and they form antigen antibody complexes. So all those antigen antibody complexes are removed by acidophils. So this is another function we can see. They also increase their number. That is, acidophils number increases whenever we suffer from any kind of Helminth's infection. Helminth, already the other day Uzma ma'am has taken a class on this, right? All roundworms that we talk about. Next, the third important type of granulocyte. See, I said granulocytes are of three types: acidophils, basophils, and neutrophils. That means neutrophils, they get stained with neutral dyes, right? So neutrophils, their number, their percentage is the highest of all, with 62%. Their population in WBCs over here is. So it is 62%. This is multi-lobe, very clear cut. The granules over here, they are small, they are abundant. They are small and they are abundant over here. These neutrophils are actually called as microscopic policemen of the body, because they are the ones, microscopic policemen of the body. What police does, our army does for us. Let's talk about army, right? They protect us from any kind of foreign invasion. So neutrophils also do the same job. On some of the neutrophil cells, some of the neutrophil cells inside the nucleus, you can find actually something called as sex chromatin body, or it is also called as drumstick body. It is called as drumstick body. What is this drumstick body? We have already learned about determination of the sex in 10th class, right? So here males have got, we have 23 set of chromosomes. So the 22 set of chromosomes are autosomes, which take care of our other characters. But the 23rd chromosome pair, which is there, it takes care of sex, right? And sex related things. So males have two chromosomes, that is X and Y, whereas females have two chromosomes, which are both the same, that is X and X. In case of females, what happens is that out of the two X chromosomes, one of the X chromosomes become inactive. It remains inactive. Only one X chromosome becomes active and it starts doing its work. So the inactivated X chromosome, it is attached to the nuclear membrane, and its appearance is just like a drumstick. That is called as sex chromatin body. So some of the neutrophils, that means sex chromatin or drumstick body is found only where X chromosome number is double. It is more than the required, which you find only in females. So if sex chromatin is present, you can understand very easily it is the condition, it is the female sex over there. That means. So this is about all the granulocytes that we have studied. Now let's talk about agranulocytes. As we can understand, A means absence of granules. These are of two types, that is monocytes and the other one being lymphocytes. Monocytes are these are large cells you can find. Nucleus is very big and it is a reniform nucleus that you can find. Reniform means what? Kidney shaped nucleus can be found over here, and cytoplasm is spread through towards the periphery. Right? Monocytes play a major role in clearing the cell debris. That means our WBCs go and attack germs. Sometimes they die, even bacteria get killed. So all that waste material that is present in the body, it is removed. So that is removed by these monocytes. Hence, these are called as scavengers of the body. Now, monocytes, when they come out of the blood and start moving in the tissues, you remember again in agranular tissues, I taught you about phagocyte cells, right? So these monocytes, when they come out of the blood, they start moving in the tissues. Some of the WBCs, once they come out, they are called as macrophages. If they are in blood, if monocytes are in blood, they are called as monocytes. These WBCs are monocytes. If they come out of the blood, then we name them as macrophages. So some of the macrophages are fixed in the tissues, and some of the macrophages are wandering type. They keep moving, searching for, if they come across any bacteria or any foreign body, they kill them. So some of them are fixed. So these are called as, the fixed ones are called as histiocytes. So this is all about monocytes. Now lymphocytes. Lymphocytes are small WBCs. They don't have granules. That is why it is under agranulocytes. Lymphocytes are actually of two types. One type of lymphocytes over here, they are called as T lymphocytes. These are T lymphocytes. The other type of lymphocytes are B lymphocytes. So these B lymphocytes are the ones which produce those antibodies. I showed you just now before, and even 2 days back also we studied about antibodies. And these are the T lymphocytes. T lymphocytes and B lymphocytes, all of them take part in protection of our body. What is the difference between them, how they protect, you will study in detail in the next year in the lesson immunology. So again, T lymphocytes over here, these are of two types. We call them as cytotoxic T cells, and these are helper T cells, TH cells and TC cells. Actually, B cells are the ones which produce antibodies, and this is the one which tries to protect our fluids, that is blood and lymph, whereas these try to protect our body in especially tissues. Okay, lifespan of WBCs. RBC's lifespan is 120 days. That means after 120 days, they die and their degradation takes place in liver and as well as in spleen. That is the reason I said liver is the cradle and the graveyard of RBCs. But when you talk about WBCs have a range, the lifespan I'm talking about, right, ranging from 1 day to many years. So some of the WBCs survive for only 2 days, 5 days, 10 days, 9 days, some half a month, and only few can survive even for years together. So they exhibit a varied range of their lifespan. So this is a slide which actually summarizes the entire information about WBCs as agranulocytes and granulocytes. Granulocytes, 3 types, right? Neutrophils, acidophils, basophils, and this one of 2 types. Okay, fine. Now another important part of blood is fluid connective tissue is a platelet. Platelets are the ones which take part in clotting of the blood. Platelets are derived from one big cell which is called as megakaryocyte. Yesterday I showed you one slide saying that bone marrow produces cells. They divide and they produce cells. Undifferentiated cells. That means their function is not determined. Afterwards, these cells, they take up their functions. So the cell which will in future become platelet, it is called as megakaryocyte. It's a big cell. And this big cell megakaryocyte, it bursts. When it ruptures, it bursts. That process is called fragmentation. And from one megakaryocyte, you find 1,000 platelets coming out. So this takes part actually in clotting of the blood, one of the most important factor. And maybe in the recent times, you would have also heard about a lot about these platelets and antibodies. During this corona time also, people were telling if at all people are there who suffered from corona and if they are having antibodies, that means antibodies are produced. So if that antibodies is given to some other patient who is suffering, he can get cured quickly. Same way, you might have heard about platelets count falling down in patients suffering from dengue and all. So there also platelets is given through in the form of injection from outside. So these two are very, very important in this way. This is a summary of entire blood, that is fluid connective tissue. This particular part I'm talking about. Lymph will start now. So blood is divided into plasma and corpuscles. We can call them as corpuscles or erythrocytes, sites otherwise. Erythrocytes, leukocytes, and blood platelets, all these things, right? Now let's move on to lymph. What is lymph? See, in the beginning, when we started connective tissue and as well as specialized connective tissue, I said there are 2 transport systems present in our body. One transport system is the famous transport system which everybody knows, that is our circulatory system, wherein heart pumps the blood, blood travels through all the parts of the body in blood vessels, that is arteries, and then it comes back to heart in veins. We know this one, right? There is accessory transport system present in our body, extra transport system present in our body, and that is called as lymphatic system. What is lymphatic system consisting of? It has got a lot of lymphoid organs. Lymphatic vessels are there carrying that lymph and lymph nodes. So here you can find that. I think I shared with you reticular connective tissue that actually provides network of support to these lymphoid organs. So some of the lymphoid organs, I already showed you this particular slide previously. So Peyer's patches in intestine, then our spleen, right? Our tonsils, all these are the places where which are all primary lymphoid organs. Secondary lymphoid organs are nothing but small nodes that are found at various places in our body. Have you seen whenever you suffer from some fever or some kind of pain, you can find swelling, small swellings in your body. So those are lymph nodes. They enlarge, they get enlarged. Why? Because the number of the WBCs over there increases to fight actually the pathogens or infecting agents. So this is the lymphatic system consisting of primary organs like spleen. These are all the primary organs. Patches in small intestine. This kills bacteria and other things. Lymph nodes are small nodules, node-like structures, and then you have this lymph vessels. So much we are talking about lymphatic vessels, lymphatic system, but first of all, what is lymph? We have to understand that. See, please try to understand this very carefully. We know that it is the artery which is carrying a pure blood, that means oxygenated blood, and as well as nutrients, supplying it to tissues, right? To tissues and to cells. So we know that through these capillaries from the arteries, blood is coming out, which is supplying nutrients and oxygen. So same blood from tissues, from cells, from body parts, from organs, it picks up carbon dioxide, and they all form capillaries of the veins that enters the vein, right? Venules, and it goes back to heart. So here you can see one main blood vessel. This is artery, which is supplying oxygenated blood. That means oxygen and nutrients to tissues over here, right? Whereas after supplying same blood, which has come up here, we know that arteries end with capillaries, right? So these capillaries, arteries end with capillaries and veins start with capillaries. So here capillaries meeting point can be seen. Arteries ending with capillaries and veins starting with capillaries, right? Meeting point can be seen over here. So this is the network of capillaries that I'm talking about, which is all this reddish in color, and these are blue capillaries. So red capillaries are supplying nutrients and oxygen to the tissues over here. After supplying that, the same blood now enters the capillaries of lymph over here, the capillaries of veins over here, and take back carbon dioxide. It takes back carbon dioxide. In this process of exchange here, you can find that the blood that has come out, plasma that has come out, nutrients, some of the molecules, some of the proteins which have come out of the capillaries of arteries, they come out because of the hydrostatic pressure, arteriolar pressure, but they can't enter the capillaries of veins. So they remain here only in these tissues, right? Now what will happen if they get removed? Nothing will happen, no? We can think like that. But every time if you find some of the plasma over here remaining, then slowly this particular tissue, or for that matter the entire body, it will swell up and finally the tissues burst, isn't it? So whatever has come out of the blood of these capillaries, whatever has come out, everything should enter into it, but a part of it gets remain here only. So as you find this is the plasma which is coming in the capillaries, and it remains, a part of it remains and part of it goes off, right? So this fluid plasma which has remained outside and did not enter veins, we call that as actually interstitial fluid or extracellular fluid. You can understand that very clearly over here all the more. So arteries supplying over here oxygen, right, and food, whereas veins take away the leftover material over here in these tissues. All that material is now picked up by another set of capillaries, that is nothing but lymphatic capillaries. So here you see the green colored capillaries, what you're seeing now. All these green colored capillaries, whatever material did not enter these veins, they take up all that material. So what is coming off over here out of the capillaries, we are saying it is blood. Actually, it is a plasma which is coming out, right? So plasma along with some of the material like proteins. All that which did not enter into these veins now start entering the capillaries of this green color structure, what you are saying. This is lymphatic capillaries. So when plasma is flowing here, we call it as blood. When this plasma comes out of the tissues, out in the tissues it is present, we call it as interstitial fluid or interstitial extracellular fluid. Extracellular fluid or interstitial fluid. Same interstitial fluid, once it enters these green capillaries, we term it as lymph. It becomes lymph. So it is blood, plasma which has come out, right? After coming out, we call it as interstitial fluid or extracellular fluid. Once it enters a green color capillaries, that is lymphatic capillaries, we call it as lymph. So lymph is nothing but it is a plasma. So whatever material did not enter veins and will not go to heart, that is picked up by these capillaries. So capillaries, these lymphatic capillaries are doing the same job what veins should be doing. Hence we say that lymphatic vessels are nothing but the parallel to venous system. This is a system which is parallel to venous system. So all that is taken back, and once again lymphatic capillaries, all of them join together, they become slightly bigger ducts, vessel capillaries, they join together to become lymphatic bigger vessels, and all of them join, still bigger ducts are formed, and finally everything is dumped back into vein itself, just before vein drops the blood into heart, that is near left subclavian vein. Now this is very, very important. See, yesterday when I was teaching you about bone, I told you that Haversian system canal, it is a central canal where you find the artery supplying blood, vein is carrying blood, and wherever these two are there, you find nerve, you find even lymphatic vessel, right? So one of the most important functions of this lymphatic capillaries is that, okay, fine, it is taking all the material which did not enter into vein, it is carrying back everything back to heart, back to blood, right? Second important function is that we know that in the small intestine, maybe you have recognized this particular slide, that this is of the folds of small intestine, right? So complete digestion takes place here in small intestine, and absorption of the food also takes place in small intestine. So here in the folds of the small intestine, you find capillaries, blood capillaries, which take the digested food, and digested food first goes to liver. Digested food first goes to liver because it is checked over there, and then goes to all parts of the body. But when fats get digested, you get fatty acids and glycerol. Fatty acids and glycerol are slightly bigger molecules. So blood capillaries don't pick them up. It is the lymphatic capillaries which carry those digested fat, that is fatty acids and glycerol. Those molecules are taken up. Digested fat is picked up by lacteals. Lacteals are the capillaries of lymphatic system. Fat is not absorbed by blood capillaries. It is taken up by lymph capillaries. So this is another second important functions of lymphatic system. So lymphatic vessels, you can understand over here very easily that arteries are supplying blood. Over here, you find this pinkish color. All this is capillaries of arteries. Then capillaries of veins, taking away that and bringing back the blood to heart. That is deoxygenated blood to heart. Whatever it got left out over here, that is picked up by these capillaries, which we call it as lymphatic capillaries, forming slightly larger ducts, and everything dumping again back into vein. Whatever these lymphatic capillaries are taking, everything is dumped back into vein itself again, near left subclavian vein. So everything enters into again system, back into the system. This is all about lymphatic vessels and lymphatic system. Then there is some other condition which is related to lymphatic system, and this is called as edema. Yesterday, maybe you remember, I told you when I was teaching about albumins and globulins that I'll teach you about one more point of albumins. We know that albumins are actually small particles which are abundantly available in blood, and one of their functions is to actually exert something called as oncotic pressure to pull back water molecule. Suppose if the albumins number goes down, then what will happen is that plasma, water comes out very easily out of the capillaries. So, and all that extra water, that is plasma, it gets remain in the interstitial fluid, what I told you earlier. But if it exceeds the limit, then you find wherever that accumulation of the plasma takes place, you find swelling in that particular region. Even we face that swelling at times. When you sit for hours together in one place, that is nothing but, it is called, that condition is called edema. Edema is the swelling of our body parts because of the accumulation of water. And where is that water coming from? That is nothing but the plasma water, which is coming from our circulatory system. Why this accumulation takes place? Because the number of the albumin gets reduced and they will not pull back water. So that condition is called edema. Now, sometimes the spelling of this edema, you may not find in some of the textbooks O. Directly EDEMA could be written. So edema condition can be found generally in older people, but even if you sit for lengthy hours or you travel for lengthy hours sitting in one place, this can develop. So if you just try to, move around every 5 minutes when you sit at a stretch, then this condition will not arise. Okay. As ma'am has in the beginning of the introduction only said that we have been talking about connective tissue derived from mesoderm, and I said that epithelial tissue is derived from all the three primary germ layers, nervous tissue is derived from ectoderm. What are these germ layers that we had been talking about? So let's learn now about it. So dear students, I'm sure that you could have already recognized these two diagrams over here. Yes, this represents the ovum, ova, the female sex gamete, and this is a sperm, right? In multicellular organisms where sexual reproduction takes place, union of these two gametes produces, the process is called as a fusion or the fertilization, and it produces zygote. Did you ever think what happens actually to this zygote over here? This zygote undergoes, now any cell is there, it undergoes division. One important point is that, ovum or ova has got only single set of chromosomes, haploid condition. Sperm also has got single set of chromosomes, right? These carry single set of chromosomes, but because of the fusion, now zygote is formed, which is diploid in nature. Because now diploid means two set of chromosomes are there, one coming from ova and the other coming from sperm, right? So now this is a diploid condition, diploid cell, zygote. What happens to this zygote? Now zygote starts dividing. It starts dividing and producing more number of cells. Now remember one thing, you have already studied in your 10th class about two types of division of cells. One is mitosis and the other one is meiosis. Because of meiosis only, sperms, sex gametes have half the number of cells, chromosomes, sorry, which is called as haploid condition. Meiotic division is responsible for that. And then we have another condition which is called as cell division called as mitosis. Did you ever think when we are born, very people could have really hold us in their hands? We were that small, but as you keep growing, what happens to your height, to the weight of your body? It keeps on increasing. Did you ever think what is responsible for that? Beyond doubt, it is the division of cells. So when one cell divides, you get two cells. That means the height will, let's imagine the height is, or the length is this much, right? Now when these two cells divide, you get four cells. So the number of cells increases. That means there is increase in the length, diameter, girth, etc. That is generally the mitotic division, how it takes place, and increases number of cells and the space. But when you talk about zygote, division of zygote, when zygote divides, yes, it forms again cells. From one cell, you get two cells. From two cells, you get four cells. When four cells divide, you get 8 cells, right? It doubles up. So 8 into 16 cells, 16 into 32, 64. When you talk about division of zygote, we don't say zygote division as division, we call it as cleavage. Now, why cleavage? Why we say it is cleavage? It is because number of cells increase over here, no doubt, but the space is not increasing, the volume is not increasing. It is the same amount of space, same amount of volume that is occupied by zygote, but the number of cells are more. That is the reason we call this as cleavage. I hope you understood what do you mean by cleavage. Cleavage means zygote dividing into number of cells. Cells are more, but the area is the same. The space occupied is the same. Now, what happens to this particular mass of the cells which gets divided over there? You find after zygote divides. So all the cells which are present over here, you find it is mass of the cells, and in appearance it is just like a mulberry fruit. Mulberry fruit. That is the reason this stage is called as morula. It is called as morula. We can compare the attachment of the cells, the adhesion of the cells, just like how you find, all the particles of bundi attached in one laddu that we eat, right? All the particles are stuck together inside and outside, everywhere around. So the morula morula stage is also like that, only with the cells attached to each other in that way, right? Later on, what happens to this morula? Now I'm talking about the embryonic development, how it takes place, how a multicellular organism is formed. That is, embryo is formed from that single cell zygote. We are talking about the various steps. Few of the steps are discussed over here. So these morula, later on, all the cells of them, they all come together and form one layer of the cell. Single layer of the cells is formed. They all get separated from each other and they all form one layer of the cells, leaving space inside. There is now space created inside. Right? The appearance of this one single layered cells with the space inside, this stage is called as blastula. This blastula stage can be compared with that of a football. Why I'm saying football is that football has inside space which we fill it up with air. So inside it is empty, which is filled up with air. Similarly, here also in blastula stage also, you find there is space inside, but this space is filled up with fluid, with water-like fluid that is released by the cells, by these cells. So this stage is called as blastula. How you find the material that makes up this football? This can be leather or whatever it is. So that is over here compared to the layer of the cells which are arranged. These cells are nothing but the cells which have come from this morula. So this is blastula stage. If you cut blastula inside, you find like this space, which is called, we can call this as blastula. We can call this as blastocyst also. And the space is called as blastocele. Blastocele. It is filled up with fluid. Blastocele is filled up with fluid, right? We can call that as primary blastocele because it is a first formed space with the first formed liquid, fluid, water-like fluid secreted, released by these particular cells. I hope you understood till here. Zygote, morula, blastula, right? What is blastula? Single layer of the cells which all accumulate inside living space. After this, what happens is that slowly, from one point, at any one point, you find that there is invagination of the cells taking place. That means these cells get pushed inside from one point. These cells, they get pushed inside. And here you can find there is one pore which develops, one hole, actually one pit is formed. What is a pit-like structure? You might have seen oranges, right? You find one depression kind of thing, right? So like that, there is one pit which develops, that is the blastopore pit. There, one hole is formed, one pore is formed over there. And slowly what happens from there is that you find some of the cells over here. Now these cells start invaginating, means getting pushed inside. Because of this, what happens is that there is a second layer of the cells which is getting developed inside. This is a second layer of the cells which is getting developed inside. And there are areas with two spaces now. There are areas with two spaces now. First space we already discussed. This is the first space which we already discussed. That is the primary blastocele, right? Blastula stage blastocele. And the second one over here is a newly developed space. This space which is developing now. This is the second space which is developing now. And there is one hole which is called over here as blastopore. Now, what about this second layer of the cells which is developing? We can say that, okay, because it has come from the same layer, from this layer should also have, in nature should be the same, like the outside layer. But it doesn't happen like that. This layer of the cells which has got pushed inside, which has got invaginated inside, they are genetically different from this layer of the cells outside. That means now this becomes the second layer of the cells, innermost layer of the cells, which is entirely different from this outside layer of the cells. The second, this layer of the cells now is called as endoderm. It is endoderm, right? Now this outside layer of the cell becomes ectoderm. This becomes the primary germ layer, first germ layer, which is ectoderm, and this becomes endoderm. So we can see the same thing over here in this particular slide. So here you can see this is the outside cell. Blastomere means they are nothing but the cells. So outside layer cells, this is ectoderm, this green layer, right? You can find there is one hole over here, which is pinkish in color. This is blastopore opening. There are two spaces. One is this space, blue color space. This is the primary space what I discussed over here. This space, and this is another space over here. Dear students, I want your kind attention towards this particular thing. This space that we are talking about here, this is actually in future it becomes digestive system. That is, this is called as archenteron. This is called as archenteron. Let's come back to this. I'll go back to those two slides once again. So this is our blastocele. This is our outermost layer, blastula stage. This is blastocele. You can find invagination of this, right? When it gets invaginated totally, the invagination starts, protruding deep inside. So this keeps moving inside, deep inside. So we get two layers of cells now. The outermost blue layer of the cells, this becomes ectoderm. This becomes ectoderm. Ectoderm gives rise to epidermis, that is the skin, nervous system, etc. And this yellow color layer, what you can see, this gives rise to endoderm. Endoderm forms digestive system, lining of the digestive system and parts of the digestive system like liver, pancreas, etc. And this space is nothing but the digestive cavity, the tract. We eat food and it goes, right? It is a pipe, hollow pipe. So this is the space of the digestive tract or gut or alimentary canal, however you may name it as. Another important thing in this diagram, you find actually this is called as archenteron. Archenteron is the primitive digestive system. Primitive digestive system in the beginning. This is embryo, right? That is why this is called as archenteron, and there is blastopore over here, right? So here we find two layers. So far we understood formation of two primary germ layers. One is this ectoderm, the other one is endoderm. Two layers are formed, and in between you have two spaces. One is this space. This space is nothing but digestive cavity space, right? Digestive tract. And this is the space that we studied over here. That is blastosphere. I hope you understood till here. Now let me go back to these two slides once again. Now, in the previous lesson when Kranti sir taught you, I'm sure you could have learned about two more important terms called as dealing with the division of zygote cells, which we call it as cleavage. One is called as radial cleavage. The other one is called as spiral cleavage. That means, see, let's first start with this. You know, this is zygote when it divided, and you have 8 cell stage and 16 like this. The cells new cells which are formed, here you find this cell placed one above the other properly, in proper way, stacked up one above the other. So if you try to, make an imaginary line passing through the center of this, you get two equal halves. You get two equal halves. Radial symmetry can be seen over here. So this is called as radial cleavage. Radial cleavage, division of the cells over here, arrangement of the cells. Whereas in spiral cleavage here, what happens is that cells are not placed exactly one above the other. So if two cells are like this, right? This is one cell, this is another cell. In between, above layer cells, they are not exactly on this. Wherever is the junction of these two cells, you find here one cell. That means junction of the lower cells, you find the above layered cells present. So that gives the appearance of a spiral shape. So when you try to make one plane passing through the center of it, you find the cells are arranged spirally. So radial cleavage can be generally seen in higher organisms like echinoderms, deuterostomes, you can find it. And spiral cleavage can be seen in protostomes. What are these protostomes and radial deuterostomes? Let us learn. So this is again the same thing. Try to make it understand. Another important striking quality is that in radial cleavage, these cells' fate is not decided. It is indeterminate. It is indeterminate. But in spiral, here you find it is determinate. That means the cells, their fate is decided. Okay, this cell will become what? This cell will become what? It is decided over here, right? Okay. Now let's talk about the third layer. So far, what I taught you over here, there are two germ layers. Development of two germ layers I finished, right? Now let's learn about the third germ layer. So again, we can see that this is the primary germ layer, ectoderm, and this was a invaginated layer, endoderm. This is the digestive cavity, and this is the pore. Just now I mentioned to you about blastopore and then something called protostomes. If this blastopore becomes mouth afterwards, then that condition is called as protostome. Protostomes, those organisms are called as protostomes. Organisms where the blastopore becomes mouth afterwards during development, such organisms are called as protostomes. And generally protostomes have this spiral cleavage. In spiral cleavage can be seen in them, right? Okay, now let's talk about the third layer. From this endoderm, this blue color layer, what you are seeing, from this endoderm, now you find few cells dividing and coming out and occupying this space, which were earlier occupied with this space. The new cells come, divide, and they occupy the space. And now you can see 3 layers. These yellow colored cells have come from this blue color cells. That is, blue color cells represent endoderm. Yellow color represents now the third new layer which is formed, which is mesoderm. Mesoderm is present in between, but it is formed afterwards. Okay, this is a speciality over here. So though this mesoderm has come from endoderm, again, as I mentioned, genetically these cells are different from these cells. So now we can see complete formation of 3 primary germ layers, ectoderm, mesoderm, and endoderm. And this is nothing but the digestive cavity with the pore over here. Hope, dear students, that you have understood the formation of 3 primary germ layers, right? Fine. Now let's move on to, so much we have discussed and learned different things, tissues, animal tissues and all. First of all, let's try to understand the different animal phyla groups that are that form part of animal kingdom over here. So we begin with the first one over here, poriferans, sponges, right? Before sponges, you have another group which are called as protozoans, where single-celled organisms are present. But earlier it was placed as separate phylum, but nowadays we study it under a group called as Protista. Under we take as 5 kingdom classification, it is studied over there. So we start animal kingdom with this poriferan sponges group, then cnidarians, hydra, jellyfishes, etc. Then platyhelminthes, flatworms, nematihelminthes, nematoda, roundworms, annelida, you find earthworms, leeches, etc. in this. Arthropoda, the largest phylum in the animal kingdom. About 80% of the animals belong to this arthropodan group, cockroaches, insects, etc. Then molluscans, these are soft-bodied animals covered with the shell either inside or outside. Shelled animals, the second largest animals group phyla in the animal kingdom. And then you find echinodermata, star-shaped starfish animals, where the skin is spiny in nature. That is the name. Sorry, that is the reason why this is called echinodermata. And finally, we have chordates, animals having backbone. So animals here are divided basically into 2 big groups. One, animals having backbone, our vertebral column that we talk about, and then animals which don't have backbone. So we find actually different animals which are around us. Around 1.8 million animal species are found, organism species are formed, right? Which we have categorized them into all these different groups. But how is it that we can learn about each and every organism? It's not like that, no? And what we try to do is that, so I'll give you one example before I proceed further. Whatever thing, if you enter into a library, right? Let us talk about library. When you enter into a library, do you find the books haphazardly, laid around everywhere on the tables? It's not like that. You find books are neatly stacked in racks. And it is very easy for us to find any book, locate any book. Why? Because books are segregated and they are placed over there. They are segregated subject-wise, author-wise, right? Topic-wise, you can find them. Alphabetical order can be found. So you can find neatly stacked. In the same way, so many different types of animals, species are found. Do you think that we can study all of them? No. And we have to neatly place them, like how you place actually the books. So there is large diversity in animal kingdom. What is this diversity? Existence of life in different shapes, different forms, living in different habitats, different conditions. We cannot study all of them. We need to somehow place the animals based on similarity in one group, based on differences in another group. So that similarities and differences, how we try to segregate organisms, we try to do using certain parameters. So those parameters, there are actually many parameters, but in our textbook, we have restricted to these 4 parameters, which are levels of organization, symmetry, cell organization, and nature of coelom. So let's try to understand each one of them one by one. So first, level of organization. When you are talking about levels of organization, we are trying to understand how the cells are placed actually. So first thing is that level of organization we study with protoplasmic level of organization. That means here, one individual cell itself acts like an individual organism. That means one cell is itself one organism. Independent organism, as how we are, multicellular organism. So that individual organism performs all the activities, respiration, breathing, everything, it takes part. It performs all those things. So this is the first level of organization. Now here in this individual organisms, we are studying about unicellular organisms, bacteria, protists, amoeba, all these things, right? Monorans and protists are covered over here. Bacteria means, of course, we know that nucleus is not distinct. This is yeast, fungus. Again, this is eukaryote, and these are all eukaryotes where nucleus is distinct. Why we say this is protoplasmic level of organization? It is because of the cytoplasm. Remember, there is difference between the word cytoplasm and protoplasm. When you're using the word cytoplasm, it means you're segregating, you're removing nucleus and nucleoplasm. When you're adding all of them, cytoplasm plus nucleus, nucleoplasm, then it becomes protoplasm. So all these functions, what go on in an individual unicellular organism, it is because of the organelles that are found over there at protoplasmic level. That is the reason this is the first level of organization. The second level of organization we talk about is multicellular organism, metazoans. Again, in this metazoans, multicellular organism, first level is cellular level of organization, which is seen in sponges, animals belonging to porifera, pore-bearing organisms. Cellular level of organization, here you can find organisms body made up of many cells. That is, these bodies are made up of many cells. They are multicellular, but tissue level is not found. They are all loose aggregate of cells. They are loosely arranged cells. That's it, can be seen over here. Functionally, the cells are different, but there is no nervous system to coordinate, control. There is no tissue, nothing. So this is called cellular level of organization. When you look at body of sponges, as I said, sponges belong to phylum Porifera. Porifera means pore-bearing organisms. That means these bodies have spores. So here, on the body surface, from the side, you find water entering inside the body, and water comes out of the body through the big opening that is found here at the top, which is called as osculum. So sponges have got two layers of cells. One layer of the cell is found outside layer of the cells, which we call it as actually pinacoderm, pinacocytes. And the inner layer of the cells, we call it as choanoderm or choanocytes. Layer is derm and cells are called as choanocytes. In between these two, there is another small layer of non-living cells and spicules and all present over here. So this picture very clearly indicates this is the body of the sponge that you can find. So these are the spaces. This is another space. These are the porocytes, which will let water inside the body. This is all empty. This is all empty where water enters from side and it goes out from here, osculum. So here you can find there is one layer of the cells outside, pinacoderm, made up of pinacocytes. The cells through which water goes inside, we call them as porocytes. And then these are the cells which are present inside, which are choanoderm, right? So these multicellular cells, you find many cells over here, but tissues are not found. So for the first time, tissue level of organization takes place in the animal kingdom. That is, in the evolution of animal kingdom, animals, we find one most important transition, and that is a formation of tissues over here. So we can see this in Cnidaria. Cnidaria, this particular phylum, old name is Coelenterata. So examples over here, you can find on your slide, hydra, jellyfishes, sea anemones, all these are the organisms belonging to this particular phyla. When we look at this particular organism, just let us try to once again keep connecting with what we have learned earlier about the germ layers. This is a body of hydra. I'm not considering this. Let us consider this, right? Hydra's body. Now we say that hydra, yes, tissues are present over here. So one tissue makes up the outer layer of the body, which we call it as epidermis. And where did this epidermis come out? Where this body stalk you are able to see, right? This outermost layer. This is epidermis, and that epidermis has come from this outermost layer. This one, ectoderm. And you can find here yellow colored layer, which is our endoderm. So here also you can find that yellow color layer, endoderm. So hydra is, we say, or for that matter, cnidarians, they are diploblastic in nature. Tissues are formed, and these tissues are arranged in the form of the layers. Two layers we can see. One is the ectoderm, the other one is the inside layer, which forms actually gastrodermis. Two layers are formed, right? Because of the presence of two layers coming from two primary germ layers, we call this as diploblastic condition. So coelentrates, all of them are diploblastic in nature. They have two primary germ layers developing in them. One is ectoderm, the other one is endoderm. So here you can see this is the blastopore, right, opening, and this is all empty space which forms the digestive tract. Same thing here, you see, this is opening, blastopore, mouth part can be opened, and this is all empty space where the food gets digested. Of course, water enters inside carrying food, right? And then you find this is the outermost layer, blue color layer. Then yellow one is the innermost layer, right? Innermost layer. So this is epidermis, inside one is gastrodermis. In between these two, can you see light colored layer, light colored? That one is nothing but this coelom that we have studied over here, this one. So outer ectoderm, inner endodermis, epidermis, outer epidermis. This is inner endodermis, which is gastrodermis here. In between these two, there is one small layer, and that small layer is this layer. So that small layer in Hydra, we call it as mesoglia. It is called as mesoglia, or mesenchymal cells are present, we say. It is non-living. That means there is no living material, no cells are formed, only fluid is present. So what is this part that we are discussing over here? That part is nothing but this digestive tract. This digestive tract. So just trying to correlate between what we have learned earlier over here and this one. That means there is only one opening. When only one opening is there, food is entering through this opening, and even undigested material is coming out through the same opening. So this is a blind sac. This digestive system is not complete. It is having only one opening. It is not having another opening wherein the unwanted material can be thrown out. So this kind of body plan, we call it as blind sac. Hope you understood the relation between this primary germ layers and this hydra's body, diploblastic condition. See, if you talk about this particular sponges also, here also you find actually two layers. One layer is outside and the other layer is inside. In between there is another layer which is mesenchymal. You find all spicules and other things. But this is not a tissue over here. That is the reason we cannot consider this condition as diploblastic condition, because they are not found forming tissues. All the cells independently they function. Function is different, but they are independent of each other. But here you find diffused network of sensory nerves trying to coordinate them. So that can be seen over here. So we understood, I hope, diploblastic condition. Next, in the level of organization, first we started with the protoplasmic level, we completed with cellular level, we completed with tissues. So we know that when cells combine, you finally get tissues, and when tissues combine, you get organs. So first level of the organs developed in the organisms called flatworms, which are platyhelminthes. Group of the organs, when they join together and perform the same task, we call it as organ system. Now, for example, in multicellular organisms like us, we find 8 organ systems present. Digestive system, which is having different organs. Mouth is one organ, food pipe is one organ, stomach is one organ, but they all work together in coordination. Their work is to digest food. Similarly, you have respiratory system, starting with nose, pharynx, trachea, etc. So like this, each you find group of organs which together perform one bigger task. Then they become organ system. So here you find development of organs and organ system, both right from platyhelminthes in animal kingdom. This is another important development in the field in evolution of animal kingdom. Okay, I taught earlier about the diploblastic condition. Now let's learn about the next germ layer, and that is triploblastic condition. You have very well understood this one, that the third layer is this middle layer, mesoderm. Some of the organisms here you can find in platyhelminthes, lower platyhelminthes, you find ectoderm, this ectoderm coming from this layer, right? Then you find over here this mesoderm, blue color cells. You can see that blue color cells is nothing but this mesoderm. How it has formed, we understood, right? Then the endoderm, the innermost one, this blue color cells, this forming the endoderm. So there are 3 germ layers present here. Animals where 3 primary germ layers are present, we call that particular organisms as triploblastic organisms. Triploblastic organisms. So from platyhelminthes onwards till end chordates, you find all the organisms having triploblastic nature. That means there are 3 primary germ layers. The body is made up of those 3 primary germ layers: ectoderm, mesoderm, and endoderm. These 3. So this space is nothing but again digestive cavity, digestive tract. So 3 germ layers are formed here: ectoderm, mesoderm, and endoderm. Let me quickly go back to one slide. I just want to show you this here. See, poriferans, only cells are found. Cnidaria, diploblastic condition. Here we have seen diploblastic, right? Hydra example. From platyhelminthes till end chordates. Chordates, only fish we have taken, but we can divide this chordates into many groups, right? And you find the highest evolved animal being us, human beings. So from platyhelminthes till the end, whatever animals you consider, all of them have triploblastic nature. That is 3 primary germ layers are found over there. Fine. After this, let's try to understand the difference once again between diploblastic and triploblastic. Dear students, I'm trying to explain this because you keep coming across this term every time you study every phyla from the next chapter onwards. So let's look at this diploblastic and triploblastic differences. So you know that we have studied this ectoderm, this is endoderm, this is digestive tract, and this is the space which is filled up with fluid, blastocele, primary blastocele. Whereas here you can find, let's not touch this. Here you can find there are 3 germ layers. This blastocele is filled up with the cells forming the 3rd layer, which is the mesoderm. It is formed afterwards, but it is present in between. You have to understand that. See, now let's try to understand the difference between diploblastic and triploblastic using these circles. So white space over here is the digestive tract, right? Digestive cavity. That is this digestive cavity I'm talking about. And over here also this digestive cavity, right? In both the conditions, white space represent digestive cavity. Surrounding digestive cavity, remember this, surrounding digestive cavity, you have always endoderm. So there, this light greenish blue color, whatever you are able to see over here and here, it is the endoderm. In both the conditions, you can say this, right? Now, let's see over here another layer. What is this layer over here? This layer in diploblastic condition, this is nothing, non-living layer. This non-living layer, what we are studying is this non-living layer. This is blastocele. Whereas here you can see orangish color layer, right? This is mesoderm over here, what you find. And outermost layers are ectoderm, blue color. So you can find the difference over here. So there is mesoderm here, there is no mesoderm. In fact, in between both the layers, you find actually only fluid material. Fluid material is this primary blastocele. Okay. Now let's move on to the next major criteria based on which we divide the organisms, right? Into, I said no, we divide the organisms into different categories. So next one, what we are going to study is about symmetry. You know what is this, right? I want to focus on imaginary axis that runs through the center of the earth, right? Primary axis around which our earth rotates. Of course, this axis is tilted. So here also, let's talk about an imaginary axis, primary axis running through the center of the earth, dividing earth into 2 equal halves. This is transverse, right? Latitude. And if you cut from top, earth, then you have longitudes, dividing the earth into 2 equal halves into right and left, right? This is superior, this is down, that is northern hemisphere and southern hemisphere, we say. Hope you understood. So what is it that I'm trying to draw your attention is that we are going to learn something called as symmetry here. So imaginary lines are used so that you can get 2 equal halves. So here symmetry means we are talking about imaginary line passing through the center of the objects. That means any line, any plane passing through the center of the shape or the object that you are taking so that you can get 2 equal halves. When you talk about symmetry, you are talking about the parts which are equal parts, which are mirror images of each other. Equal parts which are mirror images of each other. And if you discuss any axis, they are at equidistance from that primary axis that you are referring to. Then why is it that we are talking about this geometric shapes over here? There is a purpose. See, in multicellular organisms, I'm using the word again, multicellular, right? Multicellular organisms, triploblastic organisms, you find that some of the organisms, if you go through the along the primary axis, you find that some of the organs are paired. They are paired organs present. So you find them lying on either side of the primary axis at equidistance from each other, and they are mirror images of each other. So for example, over here, lungs. You have 2 lungs, one on this side and one on the other side. On both the sides, left and right side, you find lungs. Similarly, the ribs, you find 12 ribs over here, 12 ribs over here, right? So this is what is symmetry that I'm trying to say, that we are talking about the parts which are equal, which are mirror images of each other, which lie at an equidistance from the primary axis. So this is what is symmetry that I'm referring to, which exists in organisms. So how we try to define about the symmetry? Let's try to understand from lower organisms onwards, right? First, I'll start with diploblastic organisms. In diploblastic organisms, wherever diploblastic organisms are there, that means I'm talking about only 2 important phylas over here. One is Coelenterata, Cnidaria, and the other one is Ctenophora. ctenophores, we call them as comb jellies, come under that particular minor phyla. So here, when you look at this particular organism, any plane, I'm saying in any direction, that means if you make it to pass through the center of this organism, you can divide this organism into two equal halves. In any plane, you try to make any plane to pass through the center of the organism, then you can get two equal halves of this organism. Such symmetry is called as radial symmetry. This is radial symmetry. Radial symmetry can also be found actually in echinodermata, in echinoderms. But here you find, if you obviously you could have understood, I'm talking about here starfish. But here we say this is pentamerous symmetry because these organisms have 5 arms. So you can get two equal parts only in 5 different angles. You make the angle to pass through the center of this body. So this is pentamerous. But when you talk about radial symmetry, actually it is found in the adults of these echinoderms, but not in embryo during juvenile stage, larval stages. You don't find. In larval stages of echinodermata, bilateral symmetry is there. But when they become adults, you find radial symmetry. But pure radial symmetry, when you talk about, it is found in only 2 phyla. One is Coelenterata, Cnidaria, which is important, and the second one is Ctenophora. So this is a symmetry you find in here. Next, when you talk about radial symmetry, then what about the other symmetry that exists in the higher organisms? We call this as bilateral symmetry. That means only when a plane goes, only one plane in one particular direction, when you try to go through the center of the body, you can divide this organism into 2 equal halves. Not in any plane. Any plane means it is radial symmetry. Only in one plane, you can cut the organism into 2 equal halves. That is called as bilateral symmetry. It is bilateral symmetry. See, I said that in the evolution of organisms, there are first thing, tissue formation was very important. Second thing is that bilateral symmetry is very important aspect in the evolution of animals. So this is symmetry. Bilateral symmetry, only in this plane, in one plane, you can cut the organism into 2 equal halves. Only in one particular, one plane passing through the center can cut organism into 2 equal halves. So this is bilateral symmetry. Even we come under bilateral symmetry only in that heading. Asymmetrical organism, anyways, any angle you try to pass through the center, you cannot get equal parts. So this is called as asymmetrical condition. You cannot divide the organism into 2 equal halves. This is called as asymmetrical condition. Now, let's try to understand another important term, which is called as planes. Planes. You have already learned about something called as transverse section, longitudinal section, right? So let's try to understand on those lines only something called as planes. Suppose if you are, let's try to use colors to understand this. Suppose you are trying to divide, make a plane to pass through the center of the body, human being, in this way. That is orangish color you take, right? This is longitudinal. This is longitudinal division of the body. So because of this, you get 2 parts called as right and left. How many parts what you are getting? Right and left because of this division. So this is called as sagittal plane. Sagittal plane can divide the organism, that is longitudinally divide the organism into right and left parts. This is called sagittal plane. Next plane that you can see, let's talk about transverse. Take a tomato and take a knife and cut it like this. This is transverse section. In the same way, green plane, which can divide the organism into 2 parts, we call this as transverse plane. Because of this transverse plane, you get 2 types of body parts. One called as superior, above region, the other one called as inferior parts, that is below region. So this is another plane. Next, another plane that you can still divide the organism and get 2 equal parts over here is called as coronal plane, coronal, or it is also called as frontal plane. Because of this division, what happens is that we can get the ventral portion of the body and dorsal portion of the body. That means the body is cut in this way, so that we get front portion, this is ventral portion, and backside portion, that is dorsal portion. So this is how we get different planes in case of, if you study about human beings. Now let's try to understand the differences that exist in planes in other animals and us. So here you have understood very clearly, sagittal plane is longitudinal plane getting right and left parts. Transverse plane crossing, this is horizontal plane, dividing it into parts, 2 parts, superior and inferior, and the next one is showing actually the ventral and the dorsal plane. right? Frontal and coronal plane. Let's try to understand the difference with animals. See, the problem is that animals have 4 legs, right? They have 4 legs, whereas we have only 2 legs. So this portion for them, the stomach portion, this down portion facing earth, this is for them ventral surface. Where the mouth is present, this is anterior part. Where the tail is present for them, this is posterior part. This above region is dorsal surface for them. But for us, mouth and this ventral, they all are present in the same plane, right? They are all present in the same plane. So for us, where if you are talking about longitudinal axis of the body, then this becomes the anterior region and this becomes the posterior region for our body. And where all this front part is present, this is a ventral region for us. This is the anterior region or the ventral region for us, and backside becomes the posterior or the dorsal region for us. So these are the differences that you find, and the differences come because the mouth in these 4-legged animals is in front. 4-legged animals, mouth is in front clearly. But for us, head is attached to this. We are 2-legged animals, right? So you don't have particularly different area which can be called as anterior. So these are the slight differences that you find between us and animals and the planes. Okay, now coming to the most important part, dear students, and this is about coelom. Coelom means body cavity, space. Any space that is present is called as coelom or body cavity. We have already studied this earlier. I have shown you this particular slide. Why again? Can you see any space over here other than digestive system? We should not at all consider this digestive tract over here. When we talk about the space, I'm not talking about this space. I'm talking about the space that should be present over here between ectoderm and endoderm over here. There is no space at all. So 3 primary germ layers are present intact. There is no space at all. We are not going to consider this digestive tract space. So if no space is present, right, in organisms like this, then those organisms or that condition is called as acoelomate conditions, and those organisms are acoelomate organisms. No space is present at all. So those here you find all the body parts are tightly arranged because there is no space, no? But this is not an advantageous condition for organisms. Suppose you enter a room which is filled up with material. Can you walk through that? There is no space at all. So here also, for organisms, acoelomate conditions is not an advantageous situation for them because they cannot move freely, anything, their body parts inside or for their own movement also. So this is one condition. No, coelom is absent. Acoelomate means coelom is absent. Any kind of space between the germ layers is not present. No space is present within the body. No cavity is present within the body. This is acoelomate condition. So what is the next condition then? Next condition is called as pseudocoelomate. Now, one more point. Acoelomate condition is generally seen in platyhelminthes, flatworms. This condition is seen in flatworms over here. You find actually food that is present here in the digestive system. It gets digested and simply diffuses to the surrounding tissues very easily because it is no space is present. Easily the food passes from one tissue to another. That can be seen over here. Next condition is pseudocoelomate condition. Pseudocoelomate condition is seen, and this is seen in nematodes, nematihelminthes, roundworms. So Wuchereria, Ascaris, whatever examples you have studied the other day in parasites, in that particular phyla, this condition can be seen. What is seen in pseudocoelomate? Yes, let's try to understand these circles. So first you have the space, white one. This is a lumen of the gut, space of the gut, digestive cavity. We know this. This yellow color over here is the endoderm. Always digestive, the space is surrounded by endoderm. We know this, right? Then there is white space present here. Let me come back over here. Is there any space over here? There is no space. No, so acoelomate. But here you find there is some white space present, right? Coelom is present. Space means coelom is present here. Then you have one pinkish layer. This pinkish layer is of mesoderm, mesodermal layer. Mesoderm cells are present and ectoderm is there. That means, please try to listen. Pseudocoelomate is a condition where there are 3 germ layers present, right? You find from outside, let us come inside. Ectoderm is present. Surrounding ectoderm, immediately there is one layer of mesoderm. Then empty space is present. This space is filled up with primary blastocele, that coelom what we have learned earlier, no? Blastula, that coelom is present here. So ectoderm, then mesoderm, then space, then endoderm, digestive, after that digestive cavity. So space is there over here, right? Coelom is present here, but we call this coelom as pseudocoelom. Why this is called as pseudocoelom is because you find the mesoderm that is present, it is present only on one side, that is surrounding only ectoderm. See, this particular mesoderm layer which is present, this is triploblastic condition, no doubt. There are three primary germ layers, right? What are the three primary germ layers? You are fine from outside. If you come, ectoderm is present, mesoderm is present, even endoderm is present. All the three primary germ layers are present. But whatever space is present here, the space is existing between this one layer of mesoderm and endoderm. No, we call something as true coelom. In the next condition, we'll see. The mesoderm should be present on both the sides. Here, mesoderm is present only surrounding ectoderm. That is why this is called as pseudocoelom, because this is a primary coelom, blastocoelom, that you can find it over here. This pseudocoelomate for the first time can be seen in nematoda, nematohelminthes, right? Like roundworms, ascaris, whatever I mentioned. That means if you try to study this organism's body plan, you find something called as tube within tube. That means outside layer, if you say that this is ectoderm is forming one tube, outside tube, right? This is one tube, right? Then tube within tube, there is another tube. This another tube is nothing but the digestive system tube that we are talking about. That means this space. There are two spaces available here, right? So this digestive system, this empty space, this is the digestive tract that I'm referring to. This digestive space over here, which is present, is the digestive cavity over here, digestive tract over here, right? This is one tube-like structure that is existing, and another structure is the body wall itself, the main body wall. That is, this is ectoderm. Inside you find mesoderm, then you find space, then you find this is endoderm, and the space inside. The innermost space, what you're able to see is this space. The innermost space, what you're able to see over here is this space only. The wall that you're able to see over here, this wall is nothing but this yellow colored wall, endoderm. This space, what you're able to see over here is this space, the pseudocoelom space. And this surrounding that is mesoderm, pink layer, and outside you find ectoderm. This is tube within tube layer, tube within tube organization. What is the speciality of the tube within tube organization is that there is clearly a mouth, an opening through which the food enters, and there is clearly an anus through which undigested food material goes out. So there are two openings over here. You remember I told you about blastopore. So blastopore forms mouth over here, and automatically another opening develops here, by default becoming anus. So this is all about pseudocoelom. I hope you people understood this. Now let's talk about eucoelomate, true coelom. That means space is present. This space is called as true coelom, eucoelom. Eu means true. Why this is called as eucoelomate condition is that here, this is a previous pseudocoelomate condition, right? Pink layer mesoderm is found only on the ectoderm side. But here you see pink. Now let's try to understand the layers over here, circles over here. Let's come from inside. Again, this space, digestive tract, yellow color layer, endoderm, we know this. Surrounding endoderm, there is red color layer. Then there is white space. And then there is red color layer. This red color layer over here is the third layer, that is mesoderm. That means mesoderm forms lining with ectoderm. Mesoderm forms lining with endoderm, both. Mesoderm is in touch with ectoderm outside. Mesoderm is in touch with endoderm inside. Both, it is in touch with both. And then there is space over here in between. In between there is space. So hence this is called as true coelom. If at all the space is lined, cavity is lined by mesoderm on both the sides, that is endoderm and as well as ectoderm, then we call this as true coelom. So here you see, in this, what is the difference between here and here? Mesoderm is present only on one side, ectoderm. On only ectodermal side you can find mesoderm. But here mesoderm is found outside as well as mesoderm is found inside, and in between space is there. So in between mesoderm, if the space is available, cavity is available, coelom is available, then we call that condition as eucoelomate condition. We'll come to this. Now let's try to compare all the three. Let's try to compare all the three conditions. See, coelomate condition begins right from after zero, right from nematohelminthes, that is from next group, that is Annelida. From Annelida onwards, you find this triploplastic condition and eucoelomate condition. You can find over here. So there are three germ layers: ectoderm, mesoderm, endoderm. There is digestive cavity, right? And there is space within the mesoderm which is created. Splitting of the mesoderm creates the space. That is why this is called as eucoelom or trucoelom. Now let's try to understand once again the summary of all the three coeloms. All the three germ layers are there. Only this is digestive cavity, no space at all. So this is acoelomate condition, right? Now here you can find pseudocoelom. You find this is pinkish layer, mesoderm cells are present here. This is ectoderm, mesoderm, space, then endoderm, yellow color layer, endoderm. So the space is present, but this space here, it lines mesoderm only on one side. Mesoderm is present very close to ectoderm, but mesoderm is not close to endoderm. So this condition is called as pseudocoelomate condition. Now here you can see mesoderm, it forms lining with ectoderm outside. Mesoderm is present very close to endoderm. In between space is there. This is called as eucoelomate condition. So these coeloms actually derive because of the invagination of this, this gastrula, and afterwards these coeloms are formed. How coeloms are formed, let us try to understand over here. We have understood the formation of three germ layers. Now let us try to understand the formation of the coelom, how that space is created. Can you see this? See, this is the layer representing endoderm. Endoderm actually divides and gives rise to this mesoderm, which we know it fills up the complete primary blastocele, right? Complete blastocele. Now here, I just want to explain this with the help of an analogy. You find that during earthquakes, suddenly there are cracks, earth splits, the land splits, there is, and creating some cracks or space, right? Here also, once completely mesoderm is filled up over here, forming mesoderm, right? Over here, everywhere around, this mesoderm suddenly, whatever is there, it splits. So when it splits, there is space created here. So everything is mesoderm. Suddenly there is splitting of mesoderm, creating the space. Like how space is created here suddenly due to earth cracks over here. Like that, mesoderm splits and there is space created here. So if mesoderm splits and the space is created, then this kind of coelom is called as isosceles condition. You can also hear people calling it as a schizosceles condition. This is true coelom only. Schizosceles condition. Mesoderm splits and it creates space. So one layer of the mesoderm is in touch with ectoderm. Another layer of the mesoderm is in touch with endoderm and space in between. Now let's try to study another way of how this true coelom is formed. True coelom is formed. No doubt it is endoderm which produces actually this mesodermal cells. But this time space is created because of the extension of this archenteron, digestive tract. It forms out pockets. What do we mean by out pockets? Let us try to understand an analogy. See, suppose you are taking one balloon, an inflated balloon, and you start filling air. Then what happens to this balloon? Slowly it starts becoming bigger, and it gets filled up with air, right? In the same way here also, you find small out pockets. Air gets filled up here in balloons like that. This space moves into these structures over here, small structures. So there is out pocket, outgrowth or a protuberance of this archenteron forming these mesodermal cells. And this mesodermal cells, once it forms, it gets pinched off over here. This circle gets completed, it gets pinched off. So mesoderm is there, it gets pinched off with the coelom in between, and it comes and occupies over here, this side. This one also forms cell, it gets pinched off and it forms over here. So the coelom, this coelom is there, this coelom only gets enlarged. The coelom that you can find over here, this coelom gets enlarged and can be seen on the outside regions. So here the coelom, the space is present between the mesodermal cells because of the outgrowth of archenteron, the gut digestive system. This is called as enterocoelous condition, enterocoelom, and this is found in echinoderms and chordates. Hemichordates, urochordates, cephalochordates, chordates, it can be found. So once again, let's try to understand this. Here, the third layer mesoderm fills up the entire gap and mesoderm suddenly splits and the space is created. Here, mesoderm, in this condition, you find there is an outgrowth of archenteron. It forms one pocket-like structure, space enters into it, and then this one gets pinched off. When this gets pinched off together, gets separated together, there is coelom inside. So where is that coelom coming from? It is coming from this digestive tract, archenteron. So this is enterocoelous condition. So hope you understood the coelom, whether it is schizocoelous condition or enterocoelous condition. In any case, you find the same thing. You find the same thing outside ectoderm, mesoderm, then space, then mesoderm, then digestive space. So let's try to go once again. The importance of mesoderm, I'm saying, you find outside layer ectoderm, then mesoderm, then space, then again mesoderm, then digestive space. Of course, surrounding digestive space is endoderm. So mesodermal layers are of two types. One is close to ectoderm, one is close to endoderm here. The mesodermal layer which is close to ectoderm, this is called as somatic mesoderm. Mesoderm is also called otherwise as peritoneum. So somatic mesoderm can be called as parietal peritoneum, and this mesoderm which is close to digestive system is called as splanchnic mesoderm, or because it surrounds it is important part of all the visceral organs, it is also called as visceral mesoderm, visceral peritoneum otherwise. So formation of this true coelom is another important characteristic feature, landmark in the evolution of animal kingdom, right? So hope you understood this formation of this schizocoelous condition and enterocoelous condition. The same thing is explained over here in this particular also. See, ectoderm is formed, then it invaginates, endoderm is formed, and this endoderm, it forms out pockets, right? This gets sealed up and you can find here coelom. Here you can find coelom. So this is enterocoelous condition, and down this is our schizocoelous condition, what I taught. So out pockets of this archenteron giving rise to coelom is enterocoelous condition. Now, let me come back to the word what I mentioned earlier about protostomes and deuterostomes. So here we know that mouth develops, this is blastopore which develops mouth. So when this keeps on continuing over here, then another hole is formed here, pore is formed here. So for protostomes, where you find spiral cleavage. So this is mouth, then anus is formed afterwards. In deuterostomes, right? In deuterostomes, this blastopore, like echinoderms and human beings, chordates, you find blastopore becomes anus. Blastopore becomes anus and mouth is formed afterwards in deuterostomes. This is another condition. See, so here you see, this is morula stage, then blastula stage, then you find invagination, right? Gastrula stage, the pink layered cells forms mesoderm, and you can find splitting of this one, and then blastopore becoming mouth, and you find over here anus, and this is all seen in protostomes. And this is triploblastic condition where true coelom is formed. In deuterostomes, you find radial symmetry, blastopore, blastula stage, same. Here you find there is outgrowth of archenteron, right? True coelom is formed. Then here, anus is formed afterwards, right? So, dear students, please try to understand. Mesodermal, there are two layers. One mesodermal layer is intact with ectoderm. Another mesodermal layer is intact with the space inside. This is very important. And this space, what is the importance of this space? This space is very important for movement of the organs. For example, our heart, it is able to move when you jump, when you move. Lungs, they are able to move freely because of this space. So how we can try to understand is that, suppose if this is the digestive system, you find all the organs attached on the side of this. If this not digestive system, or otherwise you can take even, you can consider even the principal axis of our body. So you can find all the organs which are present, they can move freely because of the space that is available. Because of this space that is available, so the organs which are present, the wall of this glass, this tumbler represents the ectoderm, right? This is digestive system, digestive cavity, and endoderm. So whatever space is available, there you find all the organs moving freely. They can be present and moved freely. Now, in the same way, how we can compare is that suppose if you are taking the example of a pole dancer, this pole dancer can dance well only if the surrounding space is enough. Otherwise, the pole dancer cannot dance. In the same way, the space actually gives an opportunity for our organs to move freely. That is the importance. So that these organisms can move freely and there is no damage that takes place whenever they move. And here, coelom, in some of the eucoelom or true coelom that I'm talking about, in some of the organisms, it takes up some important vital roles. For example, if you're talking about cockroach, whatever space is present in the cockroach body, all that is filled up with fluid, which is called as hemolymph. That means that is doing the job of blood. The space is filled up with fluid that is doing the job of blood. So it is called as hemolymph. So coelom is very important over here, doing that particular function. And if you talk about earthworm, in earthworm, that coelom is filled up with fluid, and this is doing the job of skeleton, what our skeleton system does. So here it is called as hydrostatic skeleton. And another important question that you have with reference to this, again, mesoderm, is that you have a question called as what are retroperitoneal organs? Retroperitoneal organ means mesoderm actually covers our organs only on the ventral side. Mesoderm covers organs on both the sides. I explained you right over here in this particular thing. See, you find mesoderm attached to outside ectoderm, mesoderm attached inside also. So mesoderm forms covering of the organs. It turns into muscles and all those things. It forms. So here, mesoderm, it covers the organs, kidneys, only on the ventral side. So such organs are called as retroperitoneal organs. They are called as retroperitoneal organs. And one more point I want to just have a talk. Here you find blood, which is called as hemolymph. It is, you find in cockroaches flowing in empty spaces, sinuses we call. So blood doesn't flow in closed vessels. Whereas in us, in human beings, for example, you find the blood flowing in closed vessels, which are called as arteries, veins. So if the blood is flowing in vessels, then we call it as closed circulatory system. Otherwise, it is open circulatory system. It is called as open circulatory system. Hope with this you have understood all the three primary germ layers that we have discussed and talked about, and how we try to connect all the conditions, right? Three primary germ layers. And one important part over here. Now, here I mentioned about the paired organs lying on either side of the principal axis, right? So some of the organs which are not paired, they generally lie, in most of the conditions, they lie on the principal axis, like heart, very close to it, or like urinary bladder, sorry, lying on the median we say, median in location. That means it is on the longitudinal axis, right? So dear students, in this section, what we have understood is, if we try to understand since the beginning what we have learned, it is all about blood we have learned, right? We have learned all about blood, then types of blood cells we have learned, blood divided into RBC, WBC, platelets. We have seen all their functions. We have also understood what is lymph. Lymph is a material which doesn't enter into veins and then taken up by other stream of capillaries. Then we understood, actually, we have been learning about 3 germ layers. So how those 3 germ layers are formed, we have understood over here, right from the point of their formation, that is from zygote, then cleavage, types of 3 germ layers, how they are formed, how we divide organisms into different segments, right, into different groups, and the criteria for that, right from protoplasmic level. Then we have studied cellular level, triploblast, then tissue level of organization, organ systems, right? Then depending upon the germ layer, cellular level, diploblastic. Then based on the symmetry, we have understood, we have bilateral symmetry, radial symmetry, and asymmetrical organisms. Then finally, we also understood the planes, that differences in the planes of the animals and human beings, and what is the meaning of the term coelom, and where coelom is really helpful to us. So hope you really understood all these points. And finally, if you go through this one chart, you can understand that levels of organization, cellular body, it is found in poriferans. Then true system, that is tissues are found in organisms, starting right from cnidarians till the last. Then you find radial symmetry, then you find bilateral. So if you go through this, you can understand this is a summary of what we have learned so far. Please go through it. If you have any doubts, you can ask your concerned teachers. Thank you very much. Have a nice day and take care of yourselves.
Yeah, thank you, madam. Thanks a lot for very elaborative session. And it was little bit lengthy, but children, this is very, very important. Very, very important for your junior inter. Total, all the lessons will be based on this structural organization. Unless and until you are very strong enough to catch the terms and write down the terms and also draw a little bit, so that you'll get familiar with these terms and it becomes easy to understand the characters of all the animal kingdom. Okay, thank you very much, madam.
Ma'am, thank you very much. Entire webinar team, all the principals present over here, thank you very much. And I'm very sorry, ma'am, I extended not a little bit.
No, you have explained in detail. So that is 100% benefit for the students. So you need not mention sorry for that.
Actually, I was not.
We are very much thankful.
I was not happy with the PPT what I sent you last night.